So now is the time for the third speaker, Carla Shatz. She is a family postdoctoral professor of biology and neurobiology at the David Starr Jordan Director of BioX, Stanford University's pioneering interdisciplinary biosciences program. She received her BA in chemistry from Harvard in 1969, her master's degree from University College London as a Marshall Scholar, and her PhD from Harvard Medical School. Shatz joined the faculty at Stanford in 1978, then moved to UC Berkeley in 1992, and then to Harvard Medical School in year 2000, as chair of the Department of Neurobiology. And she returned to Stanford in 2007 to direct BioX. As all of you know, Dr. Shatz has devoted her career to unraveling the dynamic interplay between genes and environment that shapes brain circuits. I should say it's all about the very essence of our being. Shatz has earned many honors and awards, including election to the National Academy of Sciences, the American Philosophical Society, and the Royal Society of London. And quite recently, last year, she received the Gruber Neuroscience Prize. And tomorrow, the Kavli Prize. So, Carla, the floor is yours. Welcome.
I think we can go for it. So it's a great honor to be here. As the last speaker today, I would first like to thank the audience for their great enthusiasm and endurance. And I get the honor also of congratulating all of my co-laureates for a really fantastic day. I feel somehow like my brain is so full that I don't even know. I hope I can remember my own talk. But one thing, one of the messages from my talk, and I think you've already heard it, both from Dr. Marder and Dr. Mersnick, is that whether you like it or not, as long as you have been paying attention and you have not been sleeping, your brains will be different after today than they were this morning before you started. So I want to talk today about how it is that when we learn details about the developing brain and underlying mechanisms for how circuits change in the developing brain, that they turn out that they might actually be relevant for the aging brain at the other end of life. And sort of the surprise that I find myself now studying the other end of life and thinking maybe there's something we even can do about some of the devastating illnesses that occur there, such as Alzheimer's disease. But before I do that, I do want to remind everyone that neuroscience research, just like the other fields, nano and astro, that we've heard today, is a team effort. And I wouldn't be here today if it weren't for some incredible students and colleagues over the years. I've tried to find as many of their pictures as I could, thank goodness for the internet. And I'm also incredibly pleased to say that one of my academic mentors, my academic fathers, is here today, and that's Torsten Wiesel. I wish David Hubel were also with us, but sadly, David is not with us anymore. And lastly, I want to tell you about a miracle, and the miracle is that my brother John and his wife Nancy are here today, and it's a miracle because my brother suffered a major spinal cord injury several years ago, and you will see that he can walk with crutches today. And this is a triumph of mind over matter. He really enabled his own recovery through rehabilitation, And I think if we could only understand more about the underlying changes that happen during that process, and that is real neuroplasticity, we would all be better off, and there's a lot more work to be done. So my lab is really interested in how brain circuits change with use during developmental critical periods. And this is really one definition of plasticity, circuit change. And I got really interested in this question as a graduate student in David Hubel and Torsten Wiesel's lab, where they were studying the organization of the visual system, the mammalian visual system, and particularly the circuits that are responsible for binocular vision. And their work was so extraordinary and revealed almost a crystalline-like pattern of connectivity. And this is just a diagram of those connections, but I think you can also already see that the connections that come from the eyes and go to the brain are not just random, they're highly organized. And for example, the output neurons of the two eyes, which are called the retinal ganglion cells, those output neurons send visual information to the brain, both sides of the brain, and that information it begins to be combined in a structure called the LGN. But it really doesn't get directly mixed yet, in the sense that the LGN neurons here in this region get their input from the right eye, and the LGN neurons here in this region get their input from the left eye. And those form what are called eye-specific layers. So the inputs come together, but they really don't talk to each other yet. And at the next stage of information processing in the primary visual cortex, again, a very beautiful thing happens. The information relayed from the eyes to the cortex is now interdigitated, right eye, left eye, right eye, left eye, in these beautiful patches that are called ocular dominance columns. And when Hubel and Wiesel first discovered these remarkable patterns of connectivity, I think everybody thought it had to be hardwired, because if it isn't hardwired, I mean, how would you make such a pattern? The other reason that people thought it had to be hardwired is, quite remarkably, these patterns are present already before birth. They're not exact, but they're all there, including these eye-specific layers in the LGN. So how could you have experience or learning or something contribute to the formation of these connections? And so when I left the lab, I thought, well, I'd like to really study this question in more detail. And I thought a very good place to do it would be to study the development of these eye-specific layers. So they happen before birth, and it's possible to label these connections directly by injecting non-toxic tracers, like a green tracer into the left eye and a red tracer into the right eye. So we could just look and see how this beautiful eye-specific pattern emerges during prenatal life. And when we did that experiment, we got a really big surprise, because what we discovered is if we looked early enough, this pattern was not present. And in fact, the inputs from the two eyes are mixed in with each other in the lateral geniculate nucleus. So in the adult, as I said, an individual ganglion cell would send its input only to one layer, these ganglion cells to the other layer. These target LGN neurons will get functional input only from one eye, not the other. But another kind of surprising thing that we discovered is when we tested the connections early in development by actually activating the signaling from the eye to the brain, the action potential activity, we discovered that these neurons early on get functional input from both the right eye and the left eye, but later they only get input from one eye and not the other. So this is a very beautiful example of plasticity, and actually it's a beautiful example of what we call synaptic plasticity, because the connections between neurons are called synapses, and these synapses are remodeled to produce the adult pattern. And they're remodeled in two ways. Some of the connections are retained and stabilized, and some of them are actually weakened and pruned away. So both pruning and stabilization are part of the remodeling process that gives rise to the adult pattern of activity. The adult pattern of connectivity. Now, the finding that these connections really early on are both in an immature pattern mixed and that they actually work made us wonder whether function itself might be needed in order for this remodeling to happen. And the idea would be that neural activity being relayed from the eyes to the brain would drive those synapses and make them work. So we did what was technically hard, but conceptually a very easy experiment. And we blocked the activity coming from the eyes to the brain. And lo and behold, what we discovered is that the adult pattern of connectivity failed to emerge. So somehow neural activity is needed for the emergence of the adult pattern of connectivity from an immature pattern. But this was really a weird result, because where was this activity coming from? So not only do these connections form prenatally in humans or in primates or in higher mammals, cats, but even in mice, these connections form before the photoreceptors, the rods and the cones in the retina, are even working. So vision is not providing the signaling that's providing the activity for the formation of these eye-specific layers. So where is the activity coming from? So we wondered if perhaps the eyes are spontaneously generating neural activity and sending that activity to the brain. You could almost imagine like an engineer would run test patterns on circuits even before those circuits are actually used, in this case for vision. And in order to explore this idea of early spontaneous activity in the developing embryo, we removed the eyes and put them under a microscope and look to see whether the ganglion cells in the retina were actually signaling. And we used a variety of different methods to study this signaling process. And one of them is called calcium imaging, where every time a neuron becomes active and sends a signal to the brain, it actually undergoes a change in intracellular calcium. And with marvelous calcium-sensitive dyes that change in their fluorescence properties, it's possible to literally watch activity happen in the retina using this calcium imaging method. So in this little movie here, the scale is about 100 microns here to here. Every little black dot is a nerve cell. And this is all happening in the dark. The eyes are not seeing. This is spontaneously generated activity that is being made within the retina, and it's being relayed to the LGN. And this is the activity that we blocked. It's the activity that was required for the formation of the eye-specific layers. And the other thing you probably have noticed is this is the second time today you have heard about waves. I have to say a rather different length scale here. So these waves are actually waves of correlated activity in which neighboring ganglion cells, so every little dot is a ganglion cell, so you can see these waves carving out domains in the retina. And these waves are not only spontaneously active, but also provide correlations of firing between neighboring ganglion cells. And we know now that actually cells that fire together, wire together, and out of sync lose your link, in the sense that these are known cellular mechanisms of Hebbian synaptic plasticity that, in the case of synchronous activity, lead to strengthening of connections, and in the case of out of sync, lead to weakening and pruning of connections. A story for another day, but those mechanisms are here. So what I want to do now is actually ask another question, and that is a question of how is it that somehow neural activity is ultimately read out into circuit change, into the change in the structure of connections, because, right, there's this remodeling that's going on. Synapses are being strengthened or weakened. That is remodeling the circuit. That's plasticity that we're studying. And so a number of years ago, it occurred to us that maybe we could understand more about the mechanisms and molecules of this plasticity by discovering genes that might be turned on or off by the activity itself. And the way to discover those genes is to block activity or not, and then remove the nerve cells, extract the RNA, and then ask whether the levels of expression of different genes are differing depending on whether or not there's activity present. And so this is called a differential screen. And we used a method called PCR. It was a PCR-based method many years ago. And it was unbiased in the sense that we didn't know which genes we were going to find. We're just going to look for genes that are either increased or turned off when we block activity. And luckily for us, we found a few genes. One very interesting gene that is expressed at high levels. So the white means the RNA levels the gene is on, very high levels normally, and downregulated, turned down when we block neural activity. Now this was really cool, and we were very excited. And when we actually sequenced this gene to find out its identity, we were amazed to see that it was identical to a known MHC class 1 gene. This is a major histocompatibility locus class 1 gene, a mouthful. So MHC class 1. Now, this is a very large gene family. In us and in mice, there are like 50 of these genes. But we were very excited because it was clearly expressed in the healthy brain and turned down when we blocked activity. Unfortunately, we ran into a major dogma. The dogma is that the brain is immune privileged. And part of that dogma is that neurons, especially in the healthy brain, do not express these MHC class 1 proteins or genes. They turn them on maybe in damage, and also the immune cells turn them on and use them. So the dogma that we ran into is that we discovered that neurons are using these MHC class 1 genes, and they're regulated by activity in the neural circuits. So when we actually first discovered this and tried to submit our paper out, in fact, to Nature, the reviewers told us that they really respected my lab, but that we should go back to the lab and figure out what we did wrong. So this was tough, and I have to say my students have been amazing because this has happened to us a number of times. I won't go into all the gory details, but everybody's been a really intrepid explorer along with me on this journey. So we did. We went back to the lab and we worked very hard to show we were right. And this is just a working diagram of an example of one of these MHC class 1 genes. A protein, we know a number of them. Of the 50, we have some favorite ones that we know about. We know that it's in the synapse. It's near the neurotransmitter receptors, and we actually know quite a lot about this, and I'm going to come back to it in a minute. We showed that the protein, not just the RNA, but the protein is in the brain. It's in the neurons. These are neurons. It's at the synapse. It co-localizes with markers that mark the synaptic connections between the nerve cells. In fact, what happened? Yes, and so in fact, we decided we would continue and choose 2 of our favorite MHC class 1 genes, both of which we discovered were in the lateral geniculate nucleus, and ask whether they were needed for this circuit tuning property. So what do I mean by that? Well, we could ask our immunologist colleagues if we could have their mice, because these genes are required for intact immune system function. So they have made many lines of mice, transgenic and knockout mice, that lack these molecules. And so we went to our immunologist colleagues and we said, can we have your mice, please? And they said, of course you can have our mice, but they have brains, they're fine, there's nothing wrong with them. And that made us very excited. The reason we were excited is because, remember, we're studying the process of tuning up of circuits, not of the early formation of those circuits, but the tuning up of those circuits. So the mice better have brains, but then maybe the tuning up process would go awry. And so how would we look at that? Well, remember I told you that if we block activity, the eye-specific layers don't form in the LGN. So what about these knockout mice? Maybe if we injected one eye with a red tracer and another eye with a green tracer and looked in the knockout mice, we would see that they would be like the activity-blocked mice because they don't have the molecules. And remember, I told you that neural activity is needed to keep these molecules on. So without activity, there's no expression. And that's exactly what we found, in fact. In wild-type mice, we found beautiful, red eye, green eye, red eye layers. But in these knockout mice, we found large areas of the LGN that have yellow pixels indicating large areas of intermixing between the inputs, just as if it were in the immature pattern. So the mature pattern of connectivity fails to form in the mice lacking two of these MHC class 1 molecules. And not only that, but we discovered that there was also a retention functionally of extra inputs, just like the immature pattern. So the LGN neurons continue to receive these extra inputs, and they're not remodeled and pruned. Okay, so now I'm just going to say that two of these MHC molecules, the K and the D that we study, are needed both for structural and functional synapse pruning. And not only that, but it's kind of cool because we found that even though these layers do not form in the mice lacking these MHC molecules, the retinal waves are intact. So it's really amazing. What's happening is the eyes are still sending the signals to the brain, but because there are no MHC molecules, there's no way to actually translate that activity pattern into a structural or functional remodeling. So we think basically that the neuronal MHCs act downstream of neural activity, which is good because that's how we found them. We found them by blocking activity. Okay. So now let me just say that we think we're studying a synaptic system for pruning and plasticity here. But how does it really work? Well, we know the MHC class 1 molecules, some of them are involved. And we thought, well, let's take another cue from the immune system. Because in the immune system, the way these famous MHC class 1 proteins work is to present peptide to immune receptors, like the famous T-cell receptor needed for cell-mediated immunity. So what if some of these receptors are present also in the neurons? So in fact, we found through searching, we found actually quite a number of these receptors. We think we're studying the tip of the iceberg. Now, we did not find that neurons express the T-cell receptor. Whew, that's a good one because I don't want to have another big fight here. But what we did find is that neurons express, in this case, a receptor called PirB. And PirB is an innate immune receptor. And we looked for these receptors because they are expressed on other immune cells like macrophages and B cells and some dendritic cells. And they are also known to bind MHC class 1 proteins and signals. So we thought, well, what the heck, let's look for them in the neurons. PirB is a member of the Ig superfamily. It's a transmembrane receptor, and it has signaling motifs inside that transduce the binding information and do work inside the cell. And part of that work is to recruit phosphatases, which then actually break adhesion and MAP kinase cascades. And these are the very cascades that in the nervous system are required for synaptic plasticity. So we got really interested. So we found it in the brain, and then we found that it is involved in signaling that is required for synaptic plasticity, breaking synaptic plasticity actually. So how would we show that PirB is required for the kind of pruning and remodeling? Well, let's just knock it out. So again, to make many long stories short, we have generated a number of different kinds of mice that lack PirB. And what's interesting is in all of these kinds of mice, what we see is there's actually more plasticity in forebrain circuits of the knockout mice. Amazingly, there's a persistence of this exuberant juvenile-like plasticity that Mike Merznick mentioned. It persists into adult brain, and it seems to subserve actually more rapid learning and more extensive learning in adult mice. But wouldn't it be nice to have a pill? So that's what I'm thinking about at this point. So, and I just want to, point out that the protein is here, and this is in very famous nerve cells, the pyramidal cells of the neocortex in the hippocampus. So we can ask, again, how is MHC class, how is PirB working? And you should already know the answer, because we wondered if somehow in the knockout mice, pruning is different from in the wild type. Maybe synapse density is different from in the wild type. And again, to make a long story short, using methods developed in modern neuroscience labs these days, it's possible to count the number of synapses on the neocortical pyramidal cells, and you can even just choose your favorite type of pyramidal neuron. In this case, we like the ones in layer 5 of the neocortex and visual cortex. And then if we look at very high magnification, we see these little protuberances, and this is the postsynapse. So we can just count them. Big job, but very feasible. And what Maya Jurisic, a postdoc in the lab, discovered is that there are way more of these spines, these postsynapses, in the knockout mice than in the wild-type mice, at a time when a lot of pruning should have normally happened to bring the levels down. And this increase in the number of postsynapses is maintained into adulthood. So, to summarize what I've said so far, PirB is required in some way for pruning. And this is sort of interesting. The idea here is we know now, neuroscientists know, that there's a very nice dynamic balance between pruning and growth in the brain, both during development and in adulthood. And in fact, learning and memory requires some pruning and lots of growth of new connections, new synapses, because your memories are stored at synapses. So, what I've told you is that in the mouse lacking PirB, the loss of function, there is a shift, much less pruning, a bias toward growth, and that apparently creates these extra synapses, or there's failure to prune. So this is where a light bulb actually went off for us. What's the light bulb? Well, the light bulb is that this situation in PirB loss of function mice is the opposite of what happens in Alzheimer's disease. In Alzheimer's disease, there's excessive synaptic pruning. And in fact, the pruning that happens in Alzheimer's disease is thought to be driven in part by a very bad form of beta amyloid called A-beta 42, a 42 amino acid form of beta amyloid that actually clumps and oligomerizes and initially is soluble as oligomers and then becomes insoluble as the dreaded plaques that that the clinician uses to diagnose these days Alzheimer's disease. And so the question here is really, is PirB involved in this excessive synaptic pruning? And we think, at least in mice, it is, and I'm just going to say rather quickly why. So the first thing we did is we introduced our PirB knockout mice, or wild-type mice, to their mice, to the clinicians' mouse models of Alzheimer's disease. So there are many mouse models of Alzheimer's disease, but some of them are transgenic mice where the human genes known to cause early onset Alzheimer's are knocked into the mouse. And now the mouse gets Alzheimer's. So by the time it's about 9 months of age, it actually has severe memory loss. So this is a mouse that carries the sweetest mutation in APP and a deletion mutation in presenilin 1 gamma secretase. And so we crossed these mice together, and to our amazement, what we discovered is that the mice that have all of the hallmarks of Alzheimer's, in the sense that they have the plaques and they get to be 9 months of old, but if they lack PirB. So if they do not have PirB, they do not have memory loss at 9 months of age. We haven't studied them older than that, but that's amazing to me. The first question we wanted to answer is, how could this be? What's going on? What is PirB doing normally in the Alzheimer models that is somehow, if you remove it, is being protective? So again, I'm making a long story short to tell you that what we discovered is that PirB is capable of acting as a receptor for the oligomeric, the soluble oligomeric forms of beta amyloid. So what does this mean? It's a receptor. It means that it has a very clearly defined binding affinity, and we can actually measure that affinity. And what's interesting to us about it is that the affinity is an order of magnitude even higher than the normal endogenous healthy ligand for PirB. So it's almost as if these oligomers maybe acting with MHC class 1 endogenous ligands could be driving pruning in excess, for example. And so the question that we asked next is, because this is a receptor, we can really look at the downstream signaling and ask, is it consistent with the idea that it might mechanistically be driving synaptic pruning? And again, to make another long story short, I'll just say that when we studied the downstream signaling from this receptor, we discovered that in the presence of these A-beta 42 oligomers, the bad beta amyloid, we discovered that a pathway that's driving the depolymerization of actin, which is the structure needed to maintain the synapse, and that structure is being deconstructed in the presence of PirB, at least in the mice. And we are really excited about whether this might be relevant for the human condition. And we know that there are 5 members of the human PirB family called LILRBs. One of them, its N-terminal most domains also binds beta amyloid, and in human Alzheimer brain, the same disruption of that signaling pathway leading to actin depolymerization is present. So it's amazing. So what I've told you so far, and this is, I'm beginning to summarize, is that we think that at least in the mice, and maybe in the humans, Alzheimer's disease, there may be a contribution from an endogenous receptor that normally is regulated pruning, and it really gets hijacked and driven to excess by binding the beta amyloid. Okay, so now I want to kind of summarize. And here's what I want to ask, is there a pill in here? And many of you will know immediately, of course there's a pill, there's a receptor. If there's a receptor, you can target the receptor. That's what drug companies do. But, we're not a drug company, so we thought we will do a simple proof of principle. And the proof of principle was to make a kind of decoy receptor that acts like a sponge. So here's the idea. What you do is you make a recombinant protein that's only the outside of the receptor, only the extracellular domain of this receptor. And you can put it in the brain, and then it will float around the brain, and it will bind the soluble beta amyloid like a sponge, and will prevent it from binding the regular intact receptor, so the intact receptor cannot signal. So we thought, let's just do that experiment. So we actually we made the recombinant protein. So the recombinant protein is a protein. It doesn't go across the blood-brain barrier, but we can, for experiments, we can put it into little mini pumps and put it right into the brain. What should we study? Well, let's study pruning. Let's ask whether if we put this protein into an adult mouse brain, completely normal mouse, never had Alzheimer's, there's no beta amyloid there, can we actually recreate new synapses by blocking the pruning process and biasing the process toward growth? And to our real amazement, we discovered that we could, at least in the layer 5 neurons, we could increase the number of these postsynapses in the presence of this decoy receptor relative to a control infusion. Amazing to us. And not only that, but for aficionados, we also discovered that these new synapses are they're sites of functional synaptic input. Okay, so now I'm really summarizing. So here is my summary. So I just want to take a minute here, because that's how I felt. yikes. So what we've done is to increase, we created new synapses in an adult mouse brain by infusing this stuff based on our knowledge of how we think it's regulating pruning normally. And what this is telling us, I think you just heard it from Mike Merznick, is that the adult brain has an intrinsic capacity to generate many new synapses, and this is a really nice example, I think, at the cellular level and the molecular level. We're talking about the synapses here as plasticity. But there are breaks on this plasticity. And if we could understand more about the molecular logic of these breaks, it might be possible to regulate them, take them off, put them on more, in the aid of learning, cognitive enhancement, and also maybe recovery from injury. So this is my last slide, which is really, so is there an Alzheimer pill in here? We don't know, but we're very excited to try to see if we can design one. And so I think this is one aspect of the future, is to really think much more mechanistically about how this bad beta amyloid actually does its damage in the brain. We found RUN receptor. There's several other receptor candidates. And I hope in the next few years, these sort of new candidates will be added to the armamentarium of the pharmacopoeia that's being developed to treat Alzheimer's disease. And all I know is I want this to happen really soon, because I think I only have about 10 years. Okay. So with that, I just want to really summarize and remind everyone that when we started this work, we had no idea there would be any clinical relevance at all. I mean, I'm kind of shocked to even talk about this, that we started this work because we were really interested in how the eye and the brain connect up during development and how neural circuits are tuned and altered by experience. We had no idea there would be early spontaneous activity in the brain either. And we couldn't have done any of this work at all without funding agencies, increasingly these days, by the way, philanthropic, willing to take major risk on high risk but potentially, really high reward science. And I think I'm here today because it is high reward, but we didn't know that at the time. And so I really want to thank the Kavli Foundation and the award jury for the kind of recognition that they've given us, and especially to all members of my lab, because there were many, many, many days when we really wondered if we'd made the right decisions. And this is hugely important to all of us. So thank you very much.